Kevin Davies, Editing Humanity, the CRISPR Revolution and the New Era of Genome Editing. Embark on a journey through the world of genome editing and the groundbreaking CRISPR technology in the book summary of Editing Humanity, the CRISPR Revolution and the New Era of Genome Editing by Kevin Davies. Discover the history and science behind this revolutionary method that allows for precise modifications to DNA sequences, enabling us to edit the genetic code of any organism. Learn how scientists Jennifer Doudna and Emmanuelle Charpentier unlocked CRISPR's potential, and explore the implications of this powerful tool, from curing diseases to sparking ethical debates about designer babies and the future of human evolution.
A controversial discovery that shook the world of science. In 2018, He Jiankui, a Chinese scientist, used clustered regulatory interspaced palindromic repeats, CRISPR, technology to alter the genes in the cells of twin fetuses. This caused a worldwide outcry and legal issues for Jiankui. But CRISPR technology is revolutionary. It allows for quicker and easier editing of DNA than ever before. This innovation has transformed the world of science and medicine and offers the potential to cure hereditary diseases and other afflictions. Two scientists, Emmanuelle Charpentier and Jennifer Doudna, were responsible for discovering the incredible potential of CRISPR's ability to edit DNA. In 2017, two scientists used CRISPR technology to edit a gene in a human embryo. This discovery sets a new path for the future of medicine and humanity. The power of CRISPR technology. In 2000, the Human Genome Project marked a significant milestone with the sketch of the entire human genome sequence, allowing scientists to interpret the Book of Life. With this breakthrough, researchers began devising means to make useful deletions and corrections to the human genome sequence to alter the course of human evolution. This led to the advent of CRISPR technology, which offered a way to splice and edit DNA, providing a wide array of mechanisms to alter and edit DNA. Although not the first technology to edit genomes, CRISPR was a game-changer for everyone involved, immediately changing the way science is pursued. CRISPR technology now enables scientists to engineer human genes, thereby manipulating human evolution. Within a decade of decoding the genetic script, the ability to engineer changes in any organism became possible. CRISPR, the revolutionary genome editing tool. CRISPR is a portion of the bacterial genome that has been synthetically reproduced for targeted genome editing in the lab. Jennifer Doudna and Emmanuelle Charpentier revolutionized the field with their 2012 research paper, demonstrating that a CRISPR-Cas9 system could be adapted to cut almost any DNA sequence. While their results were for bacterial DNA, the potential for genetic engineering and genome editing in humans and other species is vast. The CRISPR revolution has enormous implications for new drug therapies and green revolution in chemistry. The origin of CRISPR gene editing. Jennifer Doudna and Feng Zhang were two of the primary players in the development of CRISPR gene editing. Zhang became interested in using CRISPR for genome editing, thanks to a lecture by Michael Gilmore, who realized that excessive antibiotic use disrupted the bacterial CRISPR system, leading to exposure to diseases. Zhang finally succeeded in inserting CRISPR-Cas9 into animal cells, paving the way for a new type of genetic therapy. Although Zhang continued the work done by Doudna and Charpentier, this was still a significant breakthrough in the field. These findings led to massive legal disputes about who discovered genome editing. CRISPR has the potential for entirely new forms of genetic therapy. Even though the media had yet to catch up, CRISPR was beginning to receive a lot more attention both from the media and other scientists. CRISPR milestone in cancer therapy. Carl June and his colleagues at the University of Pennsylvania Abramson Cancer Center have achieved a significant breakthrough in cancer therapy with their CRISPR trial. By using genome editing, the trial targeted T cells to locate and destroy cancer cells while preventing further destruction of T cells by cancer cells. Unlike traditional pharmaceutical treatments that only address symptoms, genome editing alters the DNA sequence, promising a permanent fix to the illness. While CRISPR's successful use still has its uncertainties, clinical trials provide reasons for hope. The latest milestone in this field has scientists and researchers excited. CRISPR and the need for ethical frameworks. The controversial journey of CRISPR continued to raise ethical concerns after Chinese scientist He Jiankui genetically edited 2 babies. The medical fraternity and society questioned the moral implications of controlling human evolution, and the consequential risks versus benefits. However, the creation of a regulatory framework for genome editing is hindered by limited expertise and comprehension of the technology. Genetics and human embryos. Gene editing in human embryos is a controversial topic that demands a well-defined ethical framework. The CRISPR-Cas9 system is a powerful tool that can be used to operate on embryo DNA, but it can also cause unintended damage. Even with the risks, advancements in research could make the procedure safer. However, discussion about gene editing often focuses on desirable traits instead of acknowledging the complexity of individual characteristics. Pre-implantation genetic testing, PGT, is already a method used to prevent problematic genes from being passed on. In cases where PGT is not an option, direct genome editing is the only choice. In the future, editing DNA in sperm or eggs before fertilization could be an alternative. The debate over gene editing raises important ethical questions that should be addressed before new technology is put into practice.
The book's summary of Editing Humanity, the CRISPR Revolution and the New Era of Genome Editing concludes with a reflection on the potential repercussions of altering the human genome. As the technology becomes increasingly refined and accessible, the potential applications and ethical concerns are immense. While CRISPR technology promises a future where we can eliminate diseases and even engineer new traits, questions surrounding safety, morality, and the extent to which we should intervene in natural processes remain. Ultimately, engaging in a global dialogue and establishing regulations to govern the use of genome editing is paramount, as we collectively navigate the profound impact of CRISPR technology on the future of humanity.